Welcome to This Explainer. Right now, as you're listening to this, you literally have trillions of bacteria peacefully coexisting inside your gut. But today, we're going to dive into a fascinating microscopic heist. We're going to uncover how one of those common bystanders orchestrates a highly coordinated, incredibly deadly attack. If you love understanding the hidden, mechanical workings of the natural world, you are absolutely in the right place.
So you gotta wonder, how does a normal, totally harmless bacteria, one that's probably living inside you and countless other animals right at this very moment, suddenly transform into a deadly pathogen? It's basically a classic Jekyll and Hyde scenario playing out on a microscopic scale. The organism we're talking about is Escherichia coli, or E. coli. And as you're about to see, the secret to its deadly alter ego lies entirely in the specific microscopic weapons it chooses to wield.
Okay, let's dive right into this. You see, shortly after any mammalian host is born, commensal E. coli basically move in from the environment. They colonize the intestinal tract. Now, these guys have very low virulence. They're just your normal flora, peacefully coexisting with the host. But then we have the rogue agents, pathogenic E. coli. The absolute defining difference between harmless coexistence and severe disease, what we in the field call Colibacillosis, is that these pathogenic strains possess specific adhesins, toxins, and capsules. It's the exact same species of bacteria, but with an entirely different, heavily armed loadout.
So, let's take a much closer look at this microscopic arsenal, the actual weapons of bacterial warfare E. coli uses against its host.
Now, the scientific term for these weapons is virulence factors. Basically, these are specific attributes that enable bacteria to colonize host mucosal surfaces and ultimately produce disease. Think of them like the highly specialized tools a cat burglar uses to break into a secure vault. Without these factors, E. coli is just an innocent bystander. But with them, it becomes a fully armed threat capable of devastating its host.
Now, what's really interesting here is how each one of these weapons plays a very specific, almost tactile role in the heist. First up, capsules. These act like microscopic stealth armor. They're made of polysaccharides, and they literally interfere with the host's immune system, stopping white blood cells from engulfing the bacteria. Then you have fimbrial adhesins. Picture these as tiny grappling hooks that allow the bacteria to firmly latch on to the intestinal walls so they don't get washed away by the gut's natural flushing action. Next come the enterotoxins, chemical sabotage signals that trick the host's own cells into rapidly leaking fluid. And finally, we have endotoxins, specifically lipopolysaccharides, which are structural pieces of the bacteria that, when released, send the host's entire systemic defense into a catastrophic, fatal overreaction.
And you know, this brilliantly illustrates just how incredibly specialized this bacterial heist really is. Because these fimbrial grappling hooks, they are absolutely not one-size-fits-all. Take the K88 adhesin, it specifically targets receptors in piglets. Meanwhile, K99 is the biological lockpick for calves and lambs. 9870P goes exclusively after neonatal piglets, and F41 targets calves. E. coli strains carry the exact specific keys for the specific biological locks of their target hosts. It's a perfect match, and it's exactly why certain strains only cause disease in very specific newborn animals.
All right, let's look at the very first major type of crime these pathogenic E. coli commit. A highly localized, totally devastating strike right at the intestinal tract, the enteric heist.
Let's move through this and see how it builds, because it's a terrifyingly rapid chain reaction. It all starts with oral infection. A newborn animal innocently ingests pathogenic E. coli from its environment. Instantly, those specialized fimbrial grappling hooks deploy, firmly attaching to the intestinal mucosa so the bacteria can't just be washed away. Once they're secured, boom, they start pumping out enterotoxins. This acts like a massive chemical hack. It forces the host's enterocytes, the intestinal cells, to hypersecrete electrolytes and fluid, while completely shutting down normal fluid absorption. The fatal result of all this? Profuse, watery diarrhea that leads to massive dehydration, severe acidosis, and tragically, often death in just a matter of days.
But hey, even within this gut attack, there are entirely different strategies at play. On one hand, you have enterotoxigenic E. coli, or ETEC. This guy is a chemical saboteur. It uses fimbria to hold on and enterotoxins to cause massive fluid loss. But here's the wild part. It causes almost zero physical damage to the cells themselves. It just rewires their instructions. On the flip side, you've got attaching and effacing E. coli, or AAEC. This one is a pure physical destroyer. It uses an adhesant called intamin to latch on, and then it physically flattens and destroys the microvilli of the host cells. This totally ruins the host's ability to absorb nutrients, leading to severe maldigestion. So it's essentially chemical hacking versus physical demolition.
But what happens when the bacteria decide the gut just isn't enough? That brings us to the blood attack, the systemic breach. The timeline for this systemic attack is incredibly urgent. Instead of just colonizing the gut, these specific strains literally break straight through the intestinal walls, the lungs, or even through the umbilical tissues of a newborn. Once they hit the bloodstream, it is game over. They multiply rapidly, using their attributes to completely shrug off the host's internal defenses. And as some of these bacteria naturally die off or get destroyed, they unleash a massive payload of endotoxins, those lipopolysaccharides from their outer membrane. This massive toxic dump triggers a violent systemic overreaction called endotoxic shock. The host's inflammatory system just goes completely into overdrive, causing hypothermia, prostration, and death, sometimes within just 24 hours.
So the crucial point to take away here is just how distinct these two battlegrounds really are. Enteric colibacillosis is a localized infection right in the intestine. It relies on fimbriae and enterotoxins to cause severe dehydration and acidosis. Colisepticaemia, on the other hand, is a rapid, aggressive invasion into the bloodstream and systemic tissues. It relies on host defense resistance and that explosive release of endotoxins to trigger shock. It is the exact same bacterial species, but completely different crimes, dictated 100% by the microscopic weapons they happen to carry.
Now, this all sounds pretty grim, right? But here's the good news. Because we understand exactly how this heist operates, we can build a highly effective defense to stop it.
Our defense entirely relies on outsmarting their weapons. The absolute ultimate countermeasure: early colostrum intake for newborn animals. Colostrum is just packed with maternal antibodies. When an animal drinks it, those antibodies actively coat the intestines and literally block those fimbrial grappling hooks from ever attaching. No attachment, no heist. Simple as that. We combine this with maintaining a clean, warm environment, which just reduces raw bacterial exposure and keeps the animal's stress levels low. We also use gradual diet changes to stop opportunistic bacteria from thriving during weaning. And finally, we use strictly targeted antimicrobials. But we only use these after susceptibility testing because, as you might guess, E. coli is notorious for evolving resistance to our drugs.
Which brings us to a perfect encapsulation of everything we've unpacked in this explainer today. Pathogenic E. coli causes Colibacillosis through strain-specific virulence mechanisms, fimbrial adhesion, enterotoxin production, attaching and effacing activity, or systemic invasion, leading principally to neonatal diarrhea or colisepticaemia in susceptible veterinary hosts. You see, it always comes back to the loadout. Without their weapons, they're just harmless passengers, but with them, deadly infiltrators. So the question I want to leave you with is this. As these bacteria continue to rapidly evolve and literally swap these weaponized traits with one another, how will our strategies need to adapt to stop the microscopic heists of tomorrow? Thanks so much for joining me on this explainer. Stay curious, and we'll see you next time.